to Cell-O-Vision, bringing you the latest in microscopic particles. We're here today with Melinda Mitosis and Greta Gamete for an interview to discuss their amazing life stories. Melinda?
Thank you, Katrina. I was born from my parent quite some time ago. I spent a long time in interphase, growing and growing in G1, until eventually I reached S phase, when my chromosomes duplicated. Then I spent a decent amount of time in G2, during which time my nuclear envelope formed and surrounded the nucleus, my chromatin duplicated, and my chromosomes, each with a pair of centrioles, duplicated as well. And before I knew it, I was ready to take part in mitosis. The process started with prophase, when my chromatin condensed into separate chromosomes. Each chromosome joined as two identical sister chromatids that were attached at the centromeres and joined together by cohesins. Also during this phase, my mitotic spindle, which is made up of centrosomes and microtubules, began to form, and the centrosomes moved away from each other. The next stage was prometaphase, when my nuclear envelope broke down and the microtubules cut through the nuclear area. Each chromatid of each chromosome now had a kinetic core, which is where some microtubules attach. The next phase, metaphase, was the longest of all the mitosis phases. The chromosomes lined up on the metaphase plate, and the kinetic cores of sister chromatids attached to kinetic core microtubules coming from opposite poles. The next phase, anaphase, was over before I knew it. My cohesins cleaved, and the two sister chromatids suddenly pulled apart and moved towards opposite ends of the cell. By the end of this phase, both ends of me had an equal number of chromosomes. One side was identical to the other, and I had twice as many chromosomes as I started with. I was now moving on to the last stage of mitosis, telophase. I suddenly contained two nuclei. My nuclear envelopes reformed, and my chromosomes were less condensed. This was the last stage of mitosis, but I still had to split into two, which happened due to cytokinesis. A cleavage furrow formed, and voila, I had a twin. It was identical not only to me, but to my parent as well. We have the same number and composition of chromosomes. This whole process was a truly momentous occasion in my life.
Thank you, Melinda, for sharing that beautiful story of your birth. Now on to Greta Gamete for another enthralling memoir.
Hi everyone! My life story began when my parents' chromosomes joined in a cell. After going through interphase and DNA replication, my parent cell entered into meiosis 1. In prophase 1, my parent cell's homologs paired with genes along the chromosome lengths as they started condensing. Unlike mitosis, synapsis occurred as my parents joined their DNA molecules by crossing over, linking hands at chiasmata to form cohesion between their sister chromatids. Now my DNA was a fourth of the new crossed-over DNA and was attached with a sister chromatid to two more sisters, a homologous chromosome pair. After the nuclear envelope broke down, the forming mitotic spindle began to move my sister and me to line up with our other two sisters at the metaphase plate. In metaphase 1, my sisters and my DNA attached to my homologous sisters by the kinetochore microtubules as we faced opposite poles. In anaphase 1, my sister and I separated from our homologous sisters as we moved to the opposite pole along the spindle. In telophase 1, half of the cell each has a haploid set of chromosomes with sister chromatids not identical to my parents. I was with my sister at one end of the cell until cytokinesis formed a cleavage furrow, separating my sister and me from our other sisters completely by making us into one of two haploid daughter cells. Our chromosomes then decondensed within a new nuclear envelope. Similar to meiosis 1, in the separation of sister chromatids, meiosis 2 occurs in both of the cells. I will tell you what happened to me while I am with my sister in one haploid cell, but my other sisters went through the same process as well. Starting with prophase 2, our two centrosomes moved toward the poles, and the mitotic spindle formed again, attached to my sisters and my chromosomes and moved us toward the metaphase plate, until in metaphase 2 I am lined up at the metaphase plate, attached to spindle microtubules by kinetochores. I am separated from my sister in anaphase 2 as we move toward opposite poles. This was the hardest part of my childhood, since I could not rely on my sister and enjoy her company. Telophase 2 is the last phase of meiosis 2, when my sister and I waited at either ends of the cell with our set of different chromosomes for cytokinesis to split us into two cells. At the same time, my other two sisters also became two cells, and together we formed four haploid daughter cells, each genetically unique from our parents. My nuclei formed around my decondensing chromosomes, and I became the gamete that I am today, with one half the number of chromosomes of my parents, and with a composition of chromosomes different from my parents and my other sisters. I think that meiosis is the best way to form offspring because it allows for genetic variation by DNA crosslinks that is crucial to evolution and generations' adaptation to changes in the environment.
Thank you, Ms. Gammy, and all of you for tuning in tonight. That concludes our show for now, but be sure to join in next week for the story of a cancer cell gone rogue. But until then, good night, everybody.